So the transfer of genetic information, what we call central dogma of biology that I've been writing over time, is simply the fact that it's simply all that is talking about gene expression. So it's just talking about gene expression. It's all about gene expression. It's gene expression. Gene expression. That is all I've been talking about. Gene expression. All right. So gene expression is what transmits transcription to produce mRNA, then translation to produce protein. All right. And we call that central dogma of biology, of genetics. Now, DNA is transcribed to RNA, and RNA is transcribed to protein, which is what I've been writing on the screen. DNA produce mRNA. DNA produce mRNA. mRNA will then produce protein. That is central dogma of biology. Right? Now, let's go. I believe I already told you that transcription happened in the DNA. Translation happened in the cytoplasm because DNA does not move out of the nucleus. So transcription happened in the nucleus. And then translation is going to happen where you have ribosome. All right? Let's go. Now, this here highlight the central dogma of biology. You see the DNA. And I was telling you on Tuesday that DNA is what determines your genotype. You can see that. Then your protein is what determines your phenotype. All right. If DNA does not convert to protein, that DNA is like a gene, and we call it recessive gene that is silent because it's not converted into protein. The DNA that is converted into protein is the DNA that is dominant. All right, let's go. So this gives you a little overview of what I talked about in transcription. I'll play it. I know you may not be able to hear the sound, but at least you will have a picture of how I told you that DNA replication, or transcription in, DNA replication rather, transcription rather in prokaryotic organism, because they have circular DNA, that it is going to be faster. So you're going to see that there. Go ahead. All right. So I'm going to highlight these few things gradually in a bit, as much as I can do before we stop. Now, we have 3 RNAs that are going to be needful here, and one of it is ribosomal RNA, call it rRNA. Its job is to make ribosome. The tRNA, we call it transfer RNA. Its job is to bring amino acid to the ribosome. And then the last one is the messenger RNA, which we call mRNA. Its job is to carry information from making specific protein from DNA to ribosome. And this is logical. Look at this. Looking at this, this is the DNA. All right. Think about the whole thing I was talking about. This is the DNA. And the DNA, I told you, central dogma of biology says information flow from DNA to mRNA. So what makes the mRNA is the DNA. And that's why it's called messenger. It's called messenger RNA because it's carrying a message. It's carrying a message. Messenger RNA because it's carrying a message. Now, if it brings the message this way, drop the message on the mRNA. So the mRNA is actually a single strand, like a smaller version of the DNA, because it's carrying the message on the DNA. What you see on mRNA is actually the message that is in DNA, but it's in a single-stranded version. And then that message from mRNA will flow from mRNA into protein. And this is the spendable version. So this is like you have a check. This is like you pay the check into your account. This is like you withdraw the cash so that you can buy gas, because nobody buy gas with check. So that's more like that. You have a check. If you pick up the check, it becomes visible for you to see. If you don't pick up the check, nobody knows you got the check. It will just be there. All right. So that is the way the system works. Now, let's go. So I will let you see a little bit of overview of translation as well. Then I'll connect it together. All right, so let's go look at this. Now, event in transcription, major thing that happens, 6 types of mRNA that is present. However, there are 3 of them that are the major ones we use. We only majorly talk about, of course, I told you about RNA primer. I told you about that earlier. We talk majorly about mRNA in transcription. We talk about rRNA, talk about tRNA. And then, of course, these guys are very important. This guy, majorly what it does, it's part of the spliceosome that is going to help to splice the DNA, the mRNA transcripts. Right, let's go. Both transcription and translation, they have the same 3 stages. However, their descriptions are different. It's just like this. When my daughter it says she have assignment. She's in she's in 1st grade. Now, if you have an assignment, you just have an assignment that I just changed now, the date. You have an assignment. She also have assignments. Now, but by the time you look at it, the content of your assignments are not the same. So even though everybody have assignments, the content of the assignments are not the same. So that's the way it is. Transcription have 3 stages: initiation, elongation, termination. Translation also have 3 stages: initiation, elongation, termination. But the details of the stages are not the same. So don't be confused and think like, oh, they have the same stage. No, they have the same terminology, but they don't have the same details. All right. Let's go. So this is transcription process. This is the initiation, the elongation, and the termination phase. Let me show you this. I think it will help. I'm trying to be fast, but all right. Now, that is the way the initiation happened, and I will show it to you here. So for transmission to happen, for initiation to happen, some few things must be in place. Number 1 is that the promoter, which is this guy here, the promoter must be able to locate, the RNA polymerase rather must be able to locate, this is the RNA polymerase, this guy, RNA polymerase, is the enzyme that wants to add the nucleotides together. It must be able to locate what we call promoter. Promoter usually has this sequence of gene, TA, so we call it, so we call it Tata box. Tata box. So usually the promoter, we always have this region, TA, is always about this. So we call it Tata box. So the promoter region is usually the Tata box. Now, RNA polymerase must first recognize the Tata box or the promoter. Now, if it does not identify the promoter, nothing will happen. So let's say this RNA polymerase has banned here before. Let's say it banned here before and it was here. This process will not produce, it will not start anything until it sees the promoter region. So it will just be running through the DNA strand, nothing will happen. It is when it sees the promoter region that production of mRNA can start. So nothing will be produced if the RNA polymerase does not identify the promoter region, which is the Tata box. So those are now another thing that must also join them is the signal factor there. Those signals that transcription can start. So those three things, this complex here must be formed. We call it initiation complex. So all of them together, we call it initiation complex. If this complex does not form, transcription cannot start. Once this forms, then the elongation process is simply that process just continue. That's all. Elongation is just like, okay, this process coupling together is initiation. The continuation of the whole thing as it runs through the strand is elongation. Termination is the other part that is significant there, which is when this guy, RNA polymerase, when it meets a termination sequence. Whenever you meet the termination sequence, we call it termination sequence, terminator, termination sequence. Whenever it meets the termination sequence, it will stop. That's it. It's just like some cars that you have now, that whenever it gets to any obstruction, it's going to stop by itself. That is the termination sequence. The car is stopping to drive, even if you want to drive more, as long as there is an obstruction. However you accelerate, the car is going to stop. That is termination sequence. All right. So those are the three stages, as simple as that. Now, okay, let's go. I'll still talk about proteins. All right. This is showing you the transcription process, how it's going to all form together to create the initiation complex and then continue the coupling just like that. All right. Okay. So at the end of the day, you are seeing the strands being made. The strand keep being made in that direction, elongation, until RNA polymerase meets this guy. It's not going to stop. When it meets this, then the termination has to happen. All right. So you see, once it meets the termination sequence, what's going to happen? The strand that has been made will be cut off. It will be dispersed. It will be removed from it. All right. Now, so transcription. RNA transcription occurs in the nucleus. I emphasized that already. Transcription occurs in mitochondria and chloroplasts as well because they have their own DNA. So they have to also undergo their own transcription. There are three types of nuclear RNA polymerases. Excuse me. So there are also different kinds of transcription factors. mRNA processed before translation. All right, now there has to be an mRNA because mRNA is the one carrying the message. It's just like this. You ask somebody to collect something from me in my office here because you are sending it to me from Houston. Now, when you are getting the stuff, if I don't get what you sent, then there's nothing that person will collect from me. All right, that's the same way the movement is, the process is. When DNA has made mRNA fully, it has delivered its message, copied it into mRNA, then mRNA can now go ahead and start translation. If transcription has not completed, translation cannot start. Now, there is a balance to that, because in prokaryotic, the both process happens one after the other, meaning that as you are transcribing, you can be translating. But of course, that area would have been made before. Yeah. Now, after in eukaryotic cell, now this statement here is majorly for the eukaryotic organism. You can see transcription differences in eukaryotic. In eukaryotic, what we make, of course, you can see what I've been writing. I write pre-mRNA, then mRNA. What we make during transcription, pre-mRNA, the pre-mRNA has to be furtherly processed. You have to process it more. The process, we call it post-transcription processing. So we call it post-transcription processes. Now, these things you are seeing here, all right, let me write here, post-transcription processes. So this part you are seeing here is, let me highlight this for you. These stages here, they are called the post-transcription processes. Number one, we call it poly A, addition of poly A cap. So we call it poly A capping. Poly A, the A is adenine. Now, sorry, this is a poly A capping here. Polyadenylation is this right here. That's a poly A capping. This here, actually, the ideal way we call it is, I mean, the wordings of the PowerPoint is great, but the ideal way we call it is like this. Actually is the top, and then we call it, we call this tail. And poly A tail, sorry, that's correct. Poly A tail, let me put it that way. Poly A tail, that's the way we call it. Then this is the G cap. Now the G cap, the G is guanine. G is guanine. A is adenine. So those are the things. And then the splicing simply means removal of the introns. I'll put that there. Please take note of that. I won't need to explain it again, because it's just a removal of introns. Now, what is intron? Introns are usually the part, removal of introns. Now, we are careful not to call it non-coding region, actually, but in the real sense, those are the parts that will not be needful in making proteins. And if they are not needful in making proteins, why keeping them anyway? So they are like excesses that will not be needful in making proteins. So they will need to be removed. So at the end of the day, after removing all of this, we call it mRNA transcript. It has been made in a way that it is just going to be making protein. Every part of it is just for making protein. Let's go. All right. I'll be rounding off soon. I just want to touch base on this. Okay, so this is what we talk about the post-transcription process. Number one, this is what we call these are the introns. The introns are the purple. The exons are the green. So exons are the one, you see, it's called protein polypeptide coding sequence or region, because they are the one that these are the one that will make protein. The green will make protein. The others, you see, non-coding region. We try not to call it not coding, just to non-coding, not to deregulate it too much, but that's the truth. It doesn't make protein. And as long as it doesn't make protein, the purpose of mRNA, the splicing is the removal of what, please? Of the introns. This will be removed. This intron, the purple, that's splicing it out, splice. So again, these are the introns. This will be removed during splicing. Splicing will remove this. Now, at the end of the day, you see what you have? You have just the red, the exon alone. Exon 1, exon 2, exon 3. All right. Now, you have a pure transcript here that is ready to make protein. So you see, the next thing is now that mRNA, this mRNA now gets out of the nucleus through the nuclear pores. All right. Because why? You don't have ribosome in the nucleus. And so mRNA need to come out of the nucleus, which is the reason for all of this dressing. You don't finish taking shower and go out naked. No, you have to dress up. So the reason for all of this GPAP, poly-A tail, and the splicing is to ensure that the ribosome is, the mRNA transit is guided out. Of course, the poly-A tail, all of that protect it and shield it, and then anchor it out of the nucleus. So that's the purpose of all of that. Let's go. Now, translation. Now, translation is pretty simple. Once I'm done with the explanation of this, then I think I will feel comfortable to stop. Now, translation is simple. Translation is this. It's like Diamelis wrote me a letter. Diamelis, do you speak Spanish? Do you Espanol? Diamelis. All right. So she's there. So, oh, okay. Now, if you had written me a letter and you wrote it in Spanish, I understand Spanish, but I mean, I don't really. The one I understand is poquito, right? I understand very little bit. I can't read it. Now, so when you write me a letter and you expect me to read it, you wrote me in Spanish. How am I going to read that letter? I will need to get somebody to read for me. The person read it for me. If the person read it in Spanish, am I going to understand it? No. So if I expect a reader, what should also be the job of that reader? Translator as well. So what I need to be able to understand what Diamedes wrote to me, thank you, is a reader and a translator, two in one. That is the same thing ribosome does to mRNA. We call what is on mRNA, we call it codon. And ribosome will just transfer that in as amino acid. So ribosome is reading codon, is reading mRNA as amino acid. And then when he reads it, it also translates it into amino acid language. It's reading it in another language. That's why we call ribosome a reader. So ribosome is a reader, all right? It's simply translating what is written in mRNA format into protein format. So here, these are what we call the genetic code. These here are the ones that when you are making it on the mRNA to make amino acid, these are the amino acids that will determine the proteins that are made. So I will show you that in a bit, but look at it. You see this UUU makes phenylalanine. UUC, phenylalanine as well. UUA make leucine, UUG make leucine. Take note of that. Now, when you come here alone, all of these four, CUU, CUC, CUA, it makes leucine. When you come here, all of these three makes isoleucine. When you come here, all of these four make valine. So we say that the code is redundant because four code can be giving rise to just one amino acid. All right. Now, again, because I won't go through the whole chapter, I will finish in about 10 minutes. The reason, so I'll be talking about some things later. It's some things that I will not get to the slide. I'm talking about majority of them now. Now, look at this. Look at this C on the isoleucine here. I mean, on the leucine here. Look at this. If you have this now, and then if there is a mutation on the third line here, if there's a mutation here, mutation simply means changes in the base, right? Mostly. If there's a mutation here that changed CUU to CUA, because it's changing the third base, there will be no change in the amino acid. And if there's no change in amino acid, there will be no functional changes. So let's say you have CUU here before, and it now changed to CUA. It is still leucine. Like here now, if you have AUC, and then the AUC change to AUA, it is still isoleucine. So whenever there's mutation on the third base, on this third base here, it most of the time does not cause any difference. So we call that kind of mutation silent mutation. We call it silent because since it's silent, it doesn't make any difference. You don't feel it, you don't know it because it doesn't have any difference. If the amino acid does not change, the function cannot change. So it doesn't change anything. So we call it silent mutation. So a mutation that is on the third base usually does not affect anything. So we call it silent mutation. I mean, there are four of the amino acid you have to take note, particularly the AUG, which is called the start codon. And we joke with it at school starts in August. And then you have the stop codon, which are three of them, UAA, UAG, UGA. Those three are stop codons. All right, let's go. So this is the same thing. You have the video here. You can look at that later. Of course, you should know that when you put your slide on presentation mode, you'll be able to play the video. All right, so translation involves this RNA here. Like I told you earlier, there is the messenger RNA, which is the one carrying the message from DNA. There's transfer RNA, which is the one that is going to bring the mRNA that should be made. And then we have the ribosomal RNA, which makes the ribosome. And that's why, so ribosome is also an RNA, all right? Okay, so we've talked about this. This here is the mRNA transcript. Oh, very good. I'm sorry, I was going to skip that. This here, no. This here, no, here I was talking about if mutation happens and the mutation affects any of these three, the third one here, you see there are three bases here, right? So if mutation happen and then it affects any of these last one, you see if a mutation change if you have CUU before, think about this with me. If you have CUU before and a mutation change the third U, this third letter, change it to CUA. What is CUU? It's leucine. What is going to be CUA? Is still leucine. Does that have any effect on your functions? No. If you have AAU, what is AAU there? Can somebody look at it and tell me, what do you think AAU is on this table? These are amino acids, right? What is AAU? AAU is asparagine. Thank you. That is AAU here, asparagine. If this AAU change to AAC, what is going to be the difference? It's still asparagine. So asparagine is ASN, is still ASN. So there's no difference. So we call that kind of mutation. That is mutation, right? But because it has no effect, we call it silent mutation. So you're going to need to study the other mutation, kind of mutations like that. Does that make sense now? I knew someone has a question. I couldn't see the name of the person. Okay,
Yes.
good.
I understood that part, but you summed it up and said if there is no changes in the amino acid, there is no changes in something, and that's what is it.
There's no change in function.
Oh, function. Okay, got it.
It's not going to have any effect on your phenotype. It's not going to have any effect on your phenotype because it has no. It's not going to affect anything because the amino acid does not change. If amino acid does not change, protein does not change. If protein does not change, no effect.
Okay, got it. Thank you.
You're welcome. All right, let me look at any other thing I can still look at before I take a break, before I stop. So if you look at this is called the mRNA transcript. This is the pre-mRNA that is made. That pre-mRNA this is modernity DNA rather. This is mRNA transcript. This is this one that will go ahead and make protein. And of course, I'll talk about this quickly before I leave. Start codon. Let's go. Let me see if I can quickly talk about the start codon. These are the ribosome subunit, different kind of subunits there. Okay, I've talked about start codon before. Now, this is the last thing I'll talk about before I go. I've illustrated that. Now, this is it. Now, this is the initiation complex for translation to happen. You see, this is your mRNA transcript. This guy carries what we call codon. And then the tRNA brings anticodon. And then the rRNA is what makes the ribosome. And these are the ribosomes here. All right. There is a large subunit. We call this large subunit. We call this small subunit. So those are the three things that makes it. And so the codons are actually read in triplets. Oops. The codons are read in triplet, like 3. So that's why you have like UAG, UUA. Those are 3. All right. So as the RNA is reading, as the ribosome is reading this mRNA transcript, it's going to be shifting it in 3. So we call that 3 reading frame. So it has a reading frame of 3. You see, the 3 is called codon. It has that 3 reading frame. So as it's reading 3, it shifts in that same manner. So it reads it and shifts, reads it and shifts. The one that has been read will shift this way. As it's read, it shifts this way, shifts this way. That is the way the ribosome does its job until it meets the transmission sequence, the termination sequence as well, which will tell the ribosome to stop making its ribosome termination sequence, to stop making protein. All right. So I think that's where I'm going to stop. Please just review the other parts. And I think I've explained some of them. Mutation is right ahead. I've talked about some of them. I review them. If you have questions, you can reach out to me though, but study and be fine. Permit me, I have to stop